What if a drug that passed every standard quality test still harmed a patient? Not because the API was wrong, not because of a formulation error, but because of a stress level impurity, something very tiny and small, your instrument literally couldn't see it. That's not a hypothetical, that's the real kind of risk that keeps analytical scientists up at night. Hi, I'm Raghuveer Singh Parmar, a pharmaceutical analytical scientist director at Revolutionized Laboratories, and today I want to talk about the technology that fundamentally changed how we approach that problem, and that is Orbitrap high resolution mass spectrometry. Why impurity profiling matters? Let's start with the why. Because if you are in pharma, you already know impurity profiling isn't optional. It's a scientific regulatory imperative. We are talking about impurities at parts per million and sometimes parts per billion concentrations, levels that seem trivial on paper but can compromise drug safety, efficacy, and your entire regulatory submission in one go. Whether you are filing with the FDA, EMA, or any other global agency, your dossier lives or dies on the quality of your impurity data. And here is the thing, impurity profiling isn't just a one-time event at the development stage. It follows the molecule throughout its entire life cycle, from early synthesis right through to commercial manufacturing. The bar doesn't drop, it gets higher and higher. Limitations of traditional techniques. So, how were we doing this before? For many years, the industry relied heavily on quadrupole systems and conventional TOF platforms. And while these instruments served their purpose, they were simply not built for the complexity of modern pharmaceutical matrices. Think about what that meant in practice. You have two compounds co-eluting from your column, their signals overlapping, and your instrument can't resolve them. One might be a known ingredient and the other completely masked, gone. So consider the reference standard dependency problem. You have to have the reference standard. You spot an unknown peak, but without a matching standard, you are stuck. You can't identify it. You are essentially flying blind, and in regulatory terms, that's the red flag. The misidentification risk alone was significant. Ambiguous data leads to incorrect structural assignments, and incorrect structural assignments lead to incorrect decisions. That's analytical risk baked into the very foundation of your workflow. Introduction to Orbitrap HRMS. This is where Orbitrap changed everything. When Orbitrap technology entered the pharmaceutical space, it wasn't just a better instrument, it was a fundamentally different way of thinking about mass spectrometry. We are talking about mass accuracy below 1 ppm level, resolving power up to 240 kFWHM. Those numbers sound abstract until you see what they mean in practice. Compounds that were previously buried inside a complex matrix now cleanly separated, precisely measured, and confidently identified. For the first time, you could determine exact molecular formulas, not approximate, not probable, but really exact. That single capability shifted the analytical conversation literally entirely. So the paradigm shift, I call this is a paradigm shift, and it's not the overstatement. Where legacy systems gave you ambiguous overlapping data and left you making interpretive guesses, the Orbitrap HRMS gives you the clarity, clean separation of co-eluting compounds, reduced matrix interferences, data that you can actually trust, and more importantly, data that you can actually defend to the regulator, to the reviewer, to a QP signing off on a release, the shift isn't just technical, it changes the confidence level of your entire analytical team. So let's talk about identifying unknown impurities. One of the most powerful and honestly most underappreciated one is the capability of Orbitrap is the ability to characterize unknown without reference standard. This is a massive advantage in the early phase drug development where standard simply doesn't exist or they are difficult to synthesize. You are seeing something in your chromatogram, you don't know what it is. With Orbitrap, you use the exact mass to assign an elemental formula. You look at the isotopic pattern to confirm it and detect any halogenation. Then you run MS-MS fragmentation and start piecing together the structure. At resolved mass, we have encountered molecules where this workflow was the only viable path forward. Complex degradation products, unexpected synthesis byproducts, situations where conventional approaches would have stalled the project entirely. The reference-free identification workflow isn't just convenient in those cases, it's what keeps the development timeline on track is our expertise. Speed meets the accuracy. Now you might be thinking all of this sounds thorough, but does it take forever? Actually, the opposite. Orbitrap platforms deliver fast scan speeds with automated data processing that compresses identification timelines very much in some workflows by up to 60%. Automated software handles peak annotation, formula assignment, and flagging, which means your scientists can spend less time doing manual review and more time doing what they're actually good at: interpretation and decision making. That's a real operational advantage, especially when you are under development pressure. Regulatory compliance advantage. From a regulatory perspective, Orbitrap data is as strong as it gets. The instrument routinely detects impurities well below the 0.1% ICH reporting threshold. We are often working at 0.05%, even 0.01%. What that kind of sensitivity means, you are not just meeting the guidelines, you are ahead of them. Your dossier becomes more comprehensive, your data package more robust, and your chances of receiving regulatory queries significantly reduced. ICH Q3A, Q3B, Q6A Orbitrap generated datasets are fully aligned with all of these FDA, EMA. The data is globally acceptable, and because the audit trail is comprehensive and traceable, it holds up under inspection. Let's talk about hybrid systems and AI. Now, where does this technology go next? Because it's not standing still. Hybrid platforms like Extractables combine targeted quantitation with untargeted screening in a single run. You are not choosing between sensitivity and discovery, you are getting both simultaneously, and increasingly machine learning algorithms are being layered on the top, accelerating peak annotation and flagging anomalies that a human reviewer might miss on pass one. Integrated with LIMS and laboratory automation, this creates an end-to-end workflow that's faster, smarter, and more traceable than anything the industry had a decade ago. We are still in an early stages of the AI-driven data interpretation will unlock, but the trajectory is clear. So to bring it together, Orbitrap isn't just a better mass spectrometer, it's a transformation in how pharmaceutical impurity science is practiced. It gives you the high confidence identification of unknowns, compliance ready datasets, faster timelines, and the analytical backbone to support quality by design across the entire drug life cycle. Any organization serious about the drug quality, whether you are a large pharma, a mid-sized generic pharma company, or a CDMO, needs to think carefully about whether this belongs in your workflow or not. The short answer in my experience is it does. If you're navigating a complex impurity challenge, dealing with unknown peaks, or preparing for a regulatory submission and want to talk through our analytical strategy, I love that conversation. Drop your questions in the comments and reach out to us directly at Resolve Mass Laboratories, because at the end of the day, the molecules don't get simpler, our sciences has to be. Thank you so much for watching this session, and see you in the next one. Till then, goodbye.